but luckily Segundo got it resolved. Oh, she just, no, let me just reply to that.
Okay.
Yeah, sorry about that. So Segundo is from Argentina. He graduated from the School of Veterinary Medicine at the Universidad de La Plata, la Facultad de Ciencias Veterinarias in Buenos Aires. So he came to work with us in June of 2019 on this large project, looking at several aspects related to transition cow, metabolism, immunology, and microbiology. But today he's going to focus on the immune component of that work, which he collaborated a lot with Dr. Driver, Dr. Nelson, and Dr. Laporte. So we're glad to have him here, and we're excited to be here to watch his presentation. With that, you can go ahead, Segundo.
Okay, well, let me go back. There you go. And I'm not good at this Zoom. Hi, everyone. Well, thank you, Chris, for the introduction. So I will start with this, this presentation that it's about the use of flow myeloid and lymphoid cell profiles to predict metritis in dairy cows. So we will start with the definition. So metritis is an acute inflammatory disease that affects the 3 layers of the uterus, and it's caused by bacteria. It's characterized by the presence of a fetid, red brownish water uterine discharge within the first 21st days postpartum. It affects around 20% of postpartum dairy cows, and it has marked consequences in the farm profitability. It also affects milk production, the reproductive performance, and because it's a painful disease, it also affects welfare. So here's just to show you the difference between a normal vaginal discharge that you can see when you go the first days postpartum, or a cow with metritis. So here is a healthy discharge. You can see that there's a normal lochia, viscous, it's clear, but this is the perfect case, right? So you can see worst case, but they are not still metritis. So you can see that they're reddish or brownish, but there's not still metritis. And here at the right, you can see this is metritis. This is the red brownish watery discharge that here, of course, it can be appreciated, but it's a really fetid discharge. So in order to characterize metritis, we use this scale with 5 different scores that was adapted from the one that was described by Chenault in 2004, in which score number 1 is the normal lochia that we just saw in the past video. That is viscous, that can be clear as we have seen, or it can be reddish or brownish. And something important is that it's not fetid and it doesn't have pus. Now, if we go to a score number 2, now you can see that this is more cloudy, it's pink, could be reddish or brownish. It's really mucoid, and it has some flecks of pus as you can see here. Now, if we go to a score number 3, you can see that this is still pink, red, or brown. It's not fetid, and that is important. And you can see that there's more pus in the discharge, but it's still less than 50%. Now, if we go to score number 4, you can see that the pus increased. Now the pus is more than 50% of the discharge, but this discharge is still not fetid, and it's not watery. And now from score 1 to 4, we consider these cows as not having metritis. And now we go to the score number 5, and this is the reddish, brownish, watery discharge. And it's really important that it's really watery, and it doesn't have pus. Sorry, please stop. What? No, I'm giving a seminar here. Wait. Let me, Clips, can you hear me? So everything is okay? Well, okay, so I have someone knocking the door saying I'm in the wrong channel. Can you hear me okay?
Yes, I can.
Okay, sorry about that. Let's keep going then. So somebody, can everyone else hear me okay? Let's see the
Seems that everybody's
chat.
replying that they can.
Yes, perfect, loud and clear. Okay, so let's keep going then. Okay, sorry about that then. So I was here in the score number 5. This is the score that we consider as having metritis. And this is the reddish brownish that is a really watery discharge, and it's fetid. And that is the most important part. So if it's not fetid, it's not metritis. So now let's continue. And as I mentioned in the first slide, metritis is diagnosed within the first 21st days postpartum. And this is the study that was done by Dr. Gavao, in which he used 753 cows, all of them with metritis, because he wanted to evaluate the frequency distribution of metritis within those 21st days postpartum. So as you can see here, 95% of all these cases will happen in the first 2 weeks postpartum. 80% of the cases will be diagnosed 10 days postpartum, and the peak incidence of metritis is 6 and 7 days after calving. Now, as I also said in the first slide, metritis has marked consequences in the farm profitability. And this is a study that was conducted by Joanny Perez-Baez, that was Dr. Calvao's last PhD student, in which their objective was to estimate the cost of metritis. They used around 12,000 cows from 16 different farms that were located in 4 different regions of the US. And as you can see here, milk production is decreased in cows that develop metritis. So by 305 days in milk, cows that develop metritis compared with non-metritic cows produce 800 kilos less milk in the entire lactation. Now, if we see the pregnancy by 305 days in milk is also decreasing cows that develop metritis when compared with non-metritic cows. And you can see that this is a 10 percentage point decrease. Now, in this study, they also collected information about the cooling, the cows that were sold, the cows that died. They also managed to get information about the dry matter intake. They calculated it with the milk production. They also collected information about the milk sales, the cow sales, the residual cow value, the feeding costs, replacement cost, cost of reproduction, treatment cost, and they came up with the conclusion that a case of metritis costs around $500. So metritis also decreases the conception rates. And in this study that was conducted by Dr. Santos, they collected health information about 5,700 cows, and they compared the conception rates between healthy cows, so with any disease, or cows with any of the diseases. And if we take a look in metritis, you can see that there's a decrease in 14% percentage points in conception rates in cows that develop metritis when we compare them with healthy cows. Now, metritis also increases pregnancy loss, and this is a graph from Rivera in 2013, in which they wanted to characterize the prevalence of peripartum diseases and their effect on reproductive performance on grazing cows. Now, if we focus here on metritis, we can see that there's an increase in pregnancy loss between 30 and 65 days after time ADI of 21%. Now, these two slides, the last 2 slides, were just to show you that metritis not only decreases pregnancy by 305 days in milk, it also decreases pregnancy rate and increases pregnancy loss. So as a consequence of this, it will also increase the intercalving interval from those cows that eventually got pregnant. Now, regarding to the cause of metritis, it is known that metritis is a multifactorial disease. So I see a way to explain this disease is with the disease triangle, in which the environment, the pathogens, and the host will play a role in the development of metritis. So there's not much has been studied about the environment, but some studies have found that, for example, metritis incidence is higher in cooler months, but others have found that the incidence increase in warmer months. And other studies found no difference between the different seasons. But now regarding to the pathogens, in this study that was conducted by Moore in 2017, they used 16S ribosomal RNA gene sequencing because they wanted to see the abundance of the different bacterial population in the uterus of virgin heifers or in different parts of the uterus from pregnant cows. So in they took samples from the amniotic fluid, the placentomes, the intercotyledonary placenta, and the cervical lumen. And what they found, as can be appreciated here, is that I think that the most important message here is that bacteria are already present in the uterus from virgin heifers. So this is way long before calving. And you can see that regarding to the proportions, you can see here that there's a 40, they found a 40% of firmicutes, a 35% of Bacteroidetes, and a 10% of Proteobacteria. So now in this study that was conducted by Jeon in 2015, in which they also used metagenomic sequencing of the 16S ribosomal RNA gene. And as you can see here, the bacterial abundance is similar between healthy and metritic cows in the day of calving up until 2 days postpartum. And after this, it deviates in cows that develop metritis in detriment of Tenoricutes and Proteobacteria, as you can see here and here, and with an increase in Bacteroidetes. They did not find, an increase in Fusobacteria, nor a change in Firmicutes. And Bicalo in 2017, he also analyzed the microbiome of the uterus, but just on the day of metritis diagnosis. And he found similar results, an increase in Bacteroidetes in cows that develop metritis, with a little increase in Fusobacteria, but a decrease in Firmicutes and Proteobacteria. Now, this increase in Fusobacteria that we saw in the study by Bicalo was also found in other studies of the uterine microbiome, and it's represented here in this figure that you can see by Dr. Gavao, in which healthy cows and cows that will develop metritis, they have the same bacterial population in the uterus by the day of calving. And then there's a switch in cows that develop metritis with an increase in Bacteroidetes and in Fusobacteria, with a decrease in Proteobacteria, Tenoricutes, and no changes in Firmicutes. Now, in contrast, cows that remain healthy, they show decreases in Bacteroidetes and in Fusobacteria, and an increase in Proteobacteria, Tenoricutes, and in Firmicutes. Now that we talked a little bit about the pathogens. Now I want to talk a little bit about the host. And for that, I want to say that there are some risk factors that are associated with metritis. For example, we have dystocia, because dystocia helps breach the physical barriers of the uterus, such as, for example, the vulva and the endometrium. Probably because it's really painful for the animal, and it will affect the dry matter intake postpartum. Another risk factor is vaginal laceration. So cows that suffer vaginal lacerations bigger than 2 centimeters have much greater incidence of metritis than cows with no laceration. And actually, this was proved in a study conducted by Achilles in 2016, in which he found that cows with laceration bigger than 2 centimeters had 63% of metritis, compared with cows with no laceration that had 35% of metritis. Now, another risk factor is primiparity, and this is probably because primipars have a higher incidence of dystocia of around 30%. Then another risk factor are stillbirth, that are also highly correlated with dystocia, and with hypocalcemia, that is another risk factor for metritis. I'm going to talk about hypocalcemia in the next few slides, so I'm not going to stop in hypocalcemia now. So another risk factors for metritis are retained placenta, uterine prolapse, and twins, and also a male calf, that are all correlated among themselves. And also retained placenta is associated with hypocalcemia. And retained placenta also may work as a fomity and carry contaminants into the vagina. Then abortion is another risk factor for metritis, not probably because of the abortion, but because of the underlying condition that caused that abortion. And also it's associated with retained placenta. And lastly, NIFAS and BHBA are risk factors for metritis. So when NIFA's prepartum are equal or higher than 0.2, sorry, not 0.2, 0.3 milliequivalents per liter, or postpartum equal or higher than 0.6 milliequivalents per liter, are associated with increased risk for metritis. The same happens with BHBA, but just postpartum, and if they are equal or higher than 10 milligrams per deciliter. Another thing that we have to have in mind regarding to metritis is that the cows are transitioning into lactation in this period, and it is well known that this period is characterized by a decrease in dry matter intake. And as you can see here, this is a study that was conducted by Hammond. You can see that cows that develop metritis have a higher decrease, a sharper decrease in dry matter intake when compared with cows that did not develop metritis. And this decrease in dry matter intake is going to cause an overall decrease in nutrients. And this within these nutrients, there's going to be a decrease in glucose, in blood glucose. And this decrease can be seen in both cows that will develop metritis and cows that won't develop metritis. And this is important because phagocytes use extracellular glucose for phagocytosis and for extracellular killing. And they also use intracellular glycogen for chemotaxis when they don't have glucose available. Now, another thing that is important that decreases, another nutrient that is important and decreases in the transition period is calcium. And as you can see here, cows that develop metritis have a sharper decrease in calcium when compared with healthy cows. This is important because ionized calcium, it's important for phagocytosis initiation and for phagosome lysosome fusion by the phagocytes. Now, another thing that is important here is that because there's a decrease in glucose, there's going to be an increase in body fat mobilization in the form of NIFA, and this increase in mobilization is higher in cows that develop metritis. And because of this increase, there's going to be an increased amount of BHBA, so an increased amount of ketone body productions by the incomplete oxidation of the NIFA in the liver, and both of them in blood have been associated with impaired neutrophil function. For example, concentrations of beta-hydroxybutyrate here, like the ones that are observed in the transition period, were shown to impair neutrophil phagocytosis, extracellular trap formation, and killing of bacteria. And also the addition of NIFA, for example, to a culture medium affected the proliferation of mononuclear cells and also the oxidative burst of neutrophils. So I think it makes sense that cows that are undergoing a severe decrease in dry matter intake, so these cows that are eating less, will have less glucose and less calcium available for the phagocytes to produce phagocytosis, and will have a higher NIFA and BHBA. And all of these probably will impair the immune cells' migration to the uterus and their ability to phagocyte microbes. And this could lead to the development of metritis. Now, here's a paper that was published by Hammond that is showing this reduction in neutrophil function because he measured cytochrome C and myeloperoxidase activity. And as you can see here, both of them are decreased in cows that develop metritis. And just to add a little bit of background, cytochrome C is used as a marker for superoxide anion, which is released by neutrophils in phagocytosis. So this is a marker for phagocytosis. And then myeloperoxidase activity, it's showing the killing capacity because myeloperoxidase, it's an enzyme that is in charge of the reaction that produces hypochlorite that kills bacteria. Now, not only the ability of the neutrophils to move to the uterus and to kill bacteria is decreased, also cows that develop metritis show a reduction in the amount of neutrophils when compared with healthy cows. And in contrast to this, the monocyte counts of cows that develop metritis are increased by the day of calving in cows that develop metritis when compared with cows that did not develop metritis. Now, despite the fact that the monocyte counts are increased, when they are stimulated with E. coli, cytokine production by the monocytes is reduced, as you can see here in these three pro-inflammatory cytokines. But something that actually my advisor told me this morning that I found really interesting about this study, which I'm not showing here, is that the cytokine production of the monocytes was increased in the day of calving in cows that develop metritis, meaning that cows that these cows already had a higher inflammatory state by the day of calving. Now, so far we have talked about the innate immune system, which, because it's the first line of defense, is the most studied for metritis. But the adaptive immune system also plays a role in the development of the immune response. So here's a study by Dr. Bromfield in 2018, in which here you can see that the CD4 positive proportion, which means that they are the T helper lymphocytes, is decreased in cows that develop metritis in the prepartum period. Not only that, the activation of these CD4 cells is increased actually in cows that develop metritis by the day of calving, because if these cells have this marker, they are not activated, they are naive. So if they lose this marker, it means they are activated. So cows that develop metritis, they have more CD4 activation by the day of calving. They also found a decrease in the CD21 proportion in cows that develop metritis. So these are the B cells. So they found a decrease in B cells in cows that develop metritis. So just to do a summary of what we saw. So by the day of calving, cows that will develop metritis present less neutrophils with lower glycogen levels, with reduced phagocytosis and killing capacity. They also show increased monocyte counts, an increased proportion of activated T helper lymphocytes, and a decrease in the proportion of B cells. And with this in mind, we came up with the hypothesis that blood myeloid and lymphoid cell profiles at calving could be predictors of metritis in dairy cows. And our objectives were to evaluate the blood myeloid and lymphoid cell profiles at calving as predictors of metritis in dairy cows. Now, regarding to the experiment, we did this experiment here in the University of Florida Dairy Research Unit from September 2019 to March 2020. We enrolled a total of 108 cows. Half of them were multipars and the other half were primipars. So basically what we did, we went in the day of calving and we collected a blood sample. We also collected information about the gestation length, the days in the close-up, the parity, the body condition score, and the calving-related disorders such as dystocia, stillbirth, vaginal laceration, etc. And we also took temperature at calving. Then we went one day after that, and we collected information about retained placenta. And then we went again 3, 7, 10, and 13 plus minus 1 days. And with a Metricheck device, we diagnosed metritis. Then with this blood that we took the day of calving, we performed flow cytometry. This is the flow cytometer that we used here in Dr. Driver's laboratory. And in here, we wanted to characterize the different leukocyte populations. And for that, we used different markers. So for myeloid cells, we use CD172 alpha. Sorry, excuse me. So there you go. We use CD172 alpha to recognize these myeloid cells. And within these myeloid cells, we use CD14 to gate monocytes. And because we didn't have a marker to differentiate the eosinophils, neutrophils, and basophils, we considered that the cells that were CD14 negative within these CD172 alpha positive cells were the granulocytes. Then for the lymphoid panel, we used to differentiate the B cells, we used MHC2 and CD21 double positive populations. Then regarding to the T lymphocytes, we wanted to divide them in gamma delta and alpha beta T cells. So regarding to the gamma delta T cells, we used gamma delta TCR. And within the alpha beta T cells, we selected the T cytotoxic cells with CD8, and the T helper cells were recognized with CD4 with the CD4 marker. So in addition to this, we also wanted to see the leukocyte activation. And for the myeloid panel, we use CD11B because it's expressed in the myeloid cells, and because it contributes to the free leukocyte addition to the endothelium. Therefore, it plays a central role in the leukocyte migration from the peripheral blood to the inflammation site. And then for the lymphoid cells, we use CD62L because it allows the lymphocytes to enter the lymphoid tissues. That is why it's called homing receptor. And because it's rapidly shed, so it's eliminated from the cell surface on cellular activation, we consider that cells, the lymphoid cells that were CD62L negative were activated cells. Now, regarding to the statistical analysis, the immune markers were analyzed with FlowJo. Then the data were analyzed with JAMP and then with SAS. In JAMP, we did an experiment correlation. We compared all the variables with Metritis, and we used a cutoff of 0.2. So the variables that had a p-value equal or lower than 0.2 were added into the model. So regarding to the model, we run it in SAS. We did a logistic regression. Our outcome was Metritis, so yes or no, because it's a categorical variable. And as covariates, we used parity, gestation length, risk factors, the days in the close-up, temperature at calving, body conditions score at calving, and immune markers with a p-value equal or lower than 0.2. Then the probability estimates from the final model were extracted from SAS. Our probability cutoff to predict Metritis was 0.42 and was determined using ROC curve in MedCalc. And then we determined sensitivity, specificity, positive and negative predictive value, and the area under the curve. Now, regarding to our results, here's the experiment correlation. So these are the variables with a p-value equal or less than 0.2, and that these are the variables that we added into the model. So as you can see, we added monocytes. This FOL is frequency of life. So this is the proportion of monocytes within the total live cells. Then we also added the myeloid cells percentage, then the frequency of live granulocytes. So again, this is the proportion of granulocytes from all the live cells. Then we added the median fluorescence intensity of the CD62L in the B cells, the median fluorescence intensity in the gamma delta T cells, the median fluorescence intensity in CD11B within the granulocytes, also the percentage of monocytes, and the difference between this percentage and this percentage of monocytes is that this one is the proportion of monocytes within the myeloid cells and the other one within the live cells. We also added activated B cells, so B cells CD62L negative percentage, and the percentage of granulocytes that were CD11B positive. So now here is the graph of the final logistic regression model to predict Metritis. And as you can see here, our significant variables were parity, risk factors, gestation length, body conditions score at calving, monocytes, and the B cells that were CD62L negative, so activated B cells. Now, regarding to parity, we had, of course, half of them were primipars, the other half were multipars. Within the primipars cows, they had 65% of Metritis. The multipars cows had 37% of Metritis. That gave us an odds ratio of 5.6, meaning that compared with multipars cows, primipars had 5.6 times higher the odds of having Metritis. Now, regarding to risk factors, within the group of cows with risk factors that were 55, they had 60% of Metritis. Cows without any risk factors that were 53 had 42% of Metritis. This gave us an odds ratio of 4, meaning that cows with a risk factor for Metritis had 4 times higher the odds of developing Metritis. Regarding to gestation length now, we had a maximum gestation length of 303 days and a minimum of 257 days. The odds ratio was 0.87. This means that each day increase in gestation length decreased the odds of developing Metritis by 15%. Now, regarding to the body condition score at calving, we had a maximum of 4.25. We use a scale that goes from in 0.25 scores up until 5. So then we had a minimum of 2.5. That gave us an odds ratio of 0.66, meaning that each 0.25 increase in the body condition score decreased the odds of developing metritis by 52%. Now, regarding to the monocytes, that this is the frequency of life. We have a maximum amount of monocytes of 10% and a minimum of less than 1%, with an odds ratio of 1.34, meaning that every 1 percentage point increase in the monocyte proportion increased the odds of developing metritis by 34%. Now, regarding to the B cell CD62L negative, we had a maximum activation of 100% and a minimum of 65%, with an odds ratio of 1.13, meaning that every 1 percentage point increase in activated B cell proportion increased the odds of developing metritis by 13%. So just to conclude, our model, including the immune markers, was able to predict metritis at calving. We had a sensitivity of 85% with a specificity of 67%. We had a positive predictive value of 71%, meaning that 71% of the cows the model predicts that are going to have metritis, they will actually develop metritis. Then we had a negative predictive value of 81%, meaning that from the cows that the model predicts that are not going to have metritis, 81% won't develop metritis. And our area under the curve was 83%. Now, after finishing this study, we started thinking why these markers were changing, and we came up with some biological possible explanations that I will tell you now. So if somebody has another opinion, I will be more than happy to, or another idea, I will be more than happy to listen to it after the seminar. But for now, this is what we came up with. And well, just as an introduction, we know that cows undergo metabolic stress. All the cows undergo metabolic stress in the transition period. And these cows that undergo metabolic stress, if they have an excessive lipid mobilization, so cows that are losing more weight than others in the transition period, will have elevated NIFAs, and this will cause more oxidative stress. This oxidative stress will increase inflammatory and immune, and will increase inflammation in general in the animal, and will cause immune dysregulation. Then this ends up becoming a vicious cycle, because these immune cells start secreting pro-inflammatory cytokines because they are activated, which is going to promote lipolysis, and then so on and so forth, right? So cows that have a higher body fat mobilization, that are going to have a higher inflammatory state, are more predisposed to develop any disease. We are, of course, trying to see if that can cause metritis, right? So we found that higher monocyte proportion in cows that develop metritis. And regarding to this, one idea that we had was about this protein that is called CCL2, that is monocyte chemioattractant protein 1, because this is a protein that this is stimulated by lipid peroxidation, and it's also upregulated following tissue injury. So this chemokine, well, regulates the migration of monocytes from the bone marrow to the bloodstream. So what we were thinking is that perhaps an excessive body fat mobilization in some animals will increase the lipid peroxidation, and this is going to cause an increased production of CCL2 by the osteoblasts, which in turn will increase the monocyte release to the bloodstream, which could cause this higher monocyte proportion in the cows that develop metritis because of a higher inflammatory state. Another thing that could be happening, because CCL2 also is upregulated within tissue injury, is that calving per se could be causing the increase in the production of CCL2, which in turn will increase the monocyte production and release to the bloodstream. So that is regarding to the monocytes. Now, if we think about the B cells, I just remember that, well, first, we found a higher B cell activation in cows that develop metritis. So cows were shedding, the B cells were shedding the CD62L. And I just want to show you in a study in which they injected CD62L positive lymphocytes to two different groups. So they injected it to uninfected mice and to salmonella infected mice. Then they collected blood samples. And what they found is that uninfected mice did not shed L-selectin in the B cells, and mice that were salmonella infected, they showed CD62L shedding in the B cells. And they also investigated which of these, which of the TLRs were in charge of this shedding. And interestingly, actually, when I was reading this study, I thought that maybe they would find that TLR4 was doing this because of LPS that is one of the PAMPs from gram-negative bacteria such as Salmonella, or maybe TLR5 because of flagellin, that's another PAMP that Salmonella has. But that was not the case, and they found that this shedding of L-selectin was mediated by TLR2, TLR3, and TLR9. So this tells us that B cells can recognize different molecules by their TLRs, and they can shed the L-selectin by doing so. So one thing that we were thinking about is that maybe DAMPs have something to do here. So DAMPs are damage-associated molecular patterns. So basically, are molecules that upon cell death are released. For example, it could be DNA, RNA, proteins, cholesterol, etc. And this can be seen here, as this is a necrotic cell that is releasing the DAMPs, right? And these are being recognized by TLRs, and these cells are mounting an inflammatory response. So these already have been described, but it's not been described in B cells. So what we were thinking is that perhaps the higher metabolic stress in cows that have more body fat mobilization will induce more cell death, and this is going to cause the release of DAMPs, and this release will bind to the TLRs in the B cells, and this is going to cause the CD62L shedding. So, and just another thing that we were searching about is that I found quite interesting is that gene ablation or inhibition of TLR2, TLR3, or TLR9 alleviates system inflammation and protects from tissue damage in these studies that were conducted in mice. So they have a role in inflammation. And they also found that B2 glycoprotein 1, RNA, and DNA that were released from these necrotic cells were suggested as potential ligands. And well, B2 glycoprotein 1 as mediated by TLR2, RNA by TLR3, and DNA by TLR9. So with that, I want to conclude this part of the seminar, and I want to thank the USDA that funded this study, and I also want to thank to Dr. Calvao, my advisor, and also to Dr. Driver, Dr. Nelson, to Jessica, Tomas, Marcela, Anna, Melanie, and Sadie, because all of them helped a lot in this study. And with that, I'm done here. So if anyone has any question or any comment or any idea, I'm more than happy to listen to it.
Perfect. Thanks for the presentation, Segundo. Open for questions. If anyone has a question, can unmute and ask, or can type in the chat if you prefer. Don't be shy. Okay,
What do you think, Lips, about this inflammation theory?
just a second.
Yes.
There's a question here.
Okay.
Given the variety of risk factors that have, from Brittany Deal, our resident, the variety of risk factors that have the potential to promote the development of metritis, do you suspect there is a considerable difference in the specific bacterial population present within the uterus of those cows depending on the specific risk factor that occurs? For example, if a cow has dystocia and another one has a vaginal tear, without dystocia, can we expect those bacterial populations to generally differ between the two based on risk factor, resulting in development of metritis and ultimately variable immune cell proportion activation? Good question, Brittany. Do you want to try to answer that, Segundo? Just,
yeah, sure, of course.
What do you think could be
So
happening?
I believe that there's not going to be any difference in the bacterial population. I think that because the bacteria are already there, what you are doing, for example, in a dystocia or in a, I don't know, in a vaginal laceration, you are opening the endometrium, and that is going to cause that those bacteria maybe start proliferating. But I believe that there's also a role of the immune system, that if that cow has a good immune system, it won't develop metritis, regardless of if she has, for example, I don't know, a vaginal laceration. But I think that basically, to sum up, no, I don't think there's going to be any difference in the bacterial population within the different risk factors.
Yeah, I think I agree with you, Segundo. And just to add a little bit more, nobody has looked at the microbiome, like classifying cows, like retained placenta, then develop metritis. Retained placenta that did not develop metritis, dystocia develop metritis, dystocia that did not develop metritis, because you need a very large sample, and you have several risk factors, right? So it becomes very challenging to stratify cows like that and still be able to, to do a meaningful analysis. Having said that, my collaborator, Rodrigo Vicalio, he did look at some aspects of the bacterial population, some specific bacteria, not some aspects, like fused bacteria, Bacteroides, in cows that had dystocia, stillbirths, and retained placenta separately, right? Looking individually, and at calving, there was no difference. So those differences happened around the time that those cows developed metritis. But again, it's just, those are major risk factors. So within that group of that had dystocia, several of those cows developed metritis. So he did not stratify dystocia, metritis, no metritis. Just looked at dystocia, just looked at everything placenta. So you cannot really separate the two. But that would have to be done, and then see if just having the risk factor and not having clinical metritis would still affect the microbiome. It could, but nobody has looked at that. Okay, there are more questions. Okay, they say that they don't have microphones, so they can type. A question from Philip.
Enjoyed your seminar, Segundo.
Thank you, Dr. Thatcher.
I was thinking, based upon some of the data that you presented and some of your proposed mechanisms, that maybe a more holistic approach is, and I think you've touched on it indirectly, that the whole oxidative system is turned on. You have a lot of oxidative stress. And I'm wondering if it's possible to manage the animals in terms of their immediate postpartum period in terms of antioxidants. selenium, feeding certain types of antioxidants that really cuts down the oxidative stress response. Your cells are there. You mentioned that cells penetrate the endometrium. The work of Dr. Archibald's kind of indicates that there is no endometrial luminal epithelium until about 34 days or so. So I'm just wondering if the entire oxidative stress phenomena is activated in these animals that really gets them into trouble, and that perhaps a dietary approach, whether it be right types of fat feeding, antioxidant materials, vitamins, selenium, particularly in Florida where you have a selenium deficient diet environment, might be a means to be able to stabilize that period as an approach. You've identified one cell type, the CD6-2L, that was highly significant. Otherwise, your other bacteria that you categorized had fairly low correlations. So I'm wondering if it's just a kind of a more holistic approach to try to change the intrauterine environment to make it having a much more antioxidative stress capability might be a possibility, and then monitor not only your incidence of your diseases, but also the activity of some of your cells that you can recover. Just a thought that was going through my mind as I was listening to you.
Yeah, I like what you're saying, and I think that if you could manage not only the supplementation, like for example of antioxidants, but also like if you really manage the prepartum and all the transition period, like not only with the diet and not only with the micro minerals, right? Because let's say you give antioxidants, but then you have cows that they are losing a lot of weight because they are in an extremely excessive negative energy balance, or probably because they are not being well treated. So I don't know up until when is it going to be efficient, the use of antioxidants. I think that we have to use it and they are going to help, but we have to take care of not only that, also all the other things, all the management and all the dietary management and the farm management, in order for the cows not to be stressed and not to stop eating before calving.
Yeah, there is data published out of the Middle East in thermogenology showing that postpartum cows that have a high oxidative status are less fertile, and that cows that if they go ahead and give a antioxidant, they get a increase in fertility.
That's it.
There's that suggestion that's out there, and it seems to me it's pretty positive area to be looking at.
That's really interesting. Yes.
One of the things that Segundo is going to look later, Dr. Thatcher, is the metabolome in blood and in the uterus. So maybe we have more insight into possible prebiotics, that can modulate the uterine microbiota. And of course, he's going to look at the microbiome as well. So that's a good and very interesting area to explore that he will explore, and hopefully we'll be able to identify something that can be used to modulate the uterine microbiome to a more positive, healthy uterus. So we have one question here, one more question in the chat from Philip. Did you find changes in rectal temperature associated with difference in cell population? And he congratulates you for the presentation as well.
Well, thank you. Thank you, Philip. And so we compared cows with metritis with healthy cows, and we did not see any difference in rectal temperature.
Hi, great presentation, Segundo. I have a few questions which are more applied for my soon-to-be seminar.
Yeah.
How can farmers benefit or apply this information/technology, and would it be economically viable for them to afford it?
Okay, so I don't think that for now, I think that this can be applied for now, and it won't be comfortable for the people in the farm. Imagine you have done flow cytometry. So imagine like giving a flow cytometry to the owner
Crash
of.
course.
Yeah, so I think this is, for now, it's just research, and this is helping us understanding what is going on. And maybe later on this, in a few years or in a 20, 30 years, this could be something that that helps in the dairy industry. But for now, it's not going to be it's not going to be practical. But for now.
Yes, based on your results, most of the correlations you found are in granulocytes and monocytes. So I was thinking, because if we use the ProCyte, which is much more easy to use for farmers, I think it would be a good approach for them, since they only have to have a sample for blood and test for monocytes and neutrophils, and they'll know everything using that machine. So maybe that's something you can look into.
Yes, I think that first we have to increase the sensitivity and specificity of our model, and maybe after that, that could be an option.
Sure. Thank you.
You're welcome.
Good question, Marcela. But at least for the monocyte percentage, yeah, you can get the ProCyte, not the markers. Not to say that, like Segundo said, that you couldn't develop a kit once you narrow down to 1 or 2 or whatever markers that could just target those and have a cheaper test. Good thinking.
Thank you.
Any others?
I just have a quick question.
Hey,
great
Fiona.
talk, Segundo. That's really nice. And I apologize that I was late for it.
Oh, that's fine.
Stuck
We
out of the
managed.
dairy.
Segundo was a hacker, and he managed to become the host. Go ahead.
So I missed the first part of your talk, but obviously I saw that the proportion of monocytes was higher in the metritic cows, but was the absolute number higher as well, or was it just the proportion?
So we did not count cells, so we don't have, sadly, we don't have that information, but I will show you here. I showed in one of our first slides that they did that, and here you can see that this is counts, and these people show that cows with metritis, they have higher monocyte counts in cows
on the day of calving.
on the day of calving, exactly.
Yep.
So it's our data shows not the same, but something really similar to them.
Yeah, that's really interesting. Thank you.
You're welcome.
Yeah, if the monocytes go up, the granulocytes go down. Yeah, makes sense. We have a difference in proportion.
I think there's another question here, Clips.
Am I sleeping? Missing questions? I don't see it.
Wait, I think I saw it. Oh yeah, Amy.
Okay.
A question about a hypothesis you made regarding your findings. You used mice models. What's the next step to test this hypothesis for cattle? Is that part of your PhD? What we want to do, it's not give an explanation like a biological explanation. We are trying to find it because it's something really interesting for us, but we want to develop a model to predict metritis. So if I stay for the PhD, what we want to do is increase the sensitivity and specificity of the model, adding metabolome and microbiome to the immune function. I don't know if anyone has any other question.
Well, if no more questions, thank you very much again, Segundo. Great presentation. And we look forward to the next seminar. I'm not sure if Fiona has any announcements or anything to add before we wrap it up.
Sure. I'll just say that I have filled the other 2 spots in the speaker schedule now. So thanks very much for those of you who expressed interest in that. And we look forward to seeing you all the rest of the semester. Thanks.
Excellent. Very good. Bye-bye then, everyone.
Perfect. Thank you very much. Bye-bye.